Thank you very much. It's a great pleasure to be here today, and actually I'm very proud to have been able to participate to this fantastic consortium. So today I'm going to give you basically a hint about how we use metabolomics to get a little bit more insight into how the microbiome, diet, and in the end how this interaction actually impacts the host metabolism. So what I'm going to show you is basically a few examples of the results we have from different studies, and I don't want to anticipate too much about other presentations, so I'm not going to show you too many data. I decided to focus really on the personal inter-individual variability because that's what we observed. That was a common point observed in all our intervention studies, is that the metabolic response is actually hugely personal. So we know already that metabolic response to diet is a mosaic of influence. But by this, I mean that we already know that the diet, the exposome, the microbiome, and our genes, we all interact to contribute to a different response in individuals. Yet, we still try to use a one-size-fits-all approach with nutrition interventions. And so the questions that we're raising here is how we move now to proper personalized nutrition. There has been a lot of studies on nutrigenetics, and we know that over 130 genes have been associated with different response in obesity. So basically, over 100 genes have been identified as partly responsible to overweight in people. Up to 70% of the variation in some studies has been associated with genes. Yet, we still can't predict individual responses to diet based on nutrigenetics. So there must be another way to do that. Well, there was a very elegant study done by the Ronald Kahn's lab that was published earlier this year that was done in mice to actually disentangle this interaction between the diet, the microbiome, and genetics. And what he did was simply to take 3 different genetic backgrounds in animals and expose them to a high-fat diet with or without antibiotics. And what he observed was that actually some metabolites, plasma metabolites, were associated specifically with genes. So the genes were the main drivers of those variations. He found that some were specific to diet, and actually in the context of a high-fat diet, he observed that the metabolites associated with fatty acids and lipid metabolism were associated with the diet, which is quite obvious. But very interestingly, he also found some patterns that were completely dependent on an interaction between genes and diet. So this illustrates that it is very difficult to predict individually the response because of the complexity of those interactions. Closer to humans, Eran Siegel's lab published a study a couple of years ago, 3 years ago now, and this was, I think it's a very famous study because it was the first one that actually demonstrated in humans that we could predict the glycemic response, so the postprandial glucose response following a meal, so a very simple diet intake. In this case, he compared, as you can see, I can't really show, it's not very, maybe on that side. As you can see on this part of the slide, you had 2 exposures, either banana here or cookies, or glucose and bread, and he shows that glucose and bread tend to have similar responses in these 2 individuals. But when you look at banana and cookies, everybody would anticipate that a banana releases very slowly the glucose, and therefore you wouldn't expect a peak of glucose following a banana intake. On the other way, you would expect a cookie to provoke a very high increase in glucose. And he saw that in some individuals, that was exactly the opposite. The banana was the one that induced the very high peak of glucose and not the cookie. So this person doesn't respond in an anticipated way or an expected way to commonly used pieces of food. And he found that the predictors for those different responses were actually quite complex. It was a combination of BMI, systolic blood pressure was one of the predictors, ALT, glycated hemoglobin, and also some bacteria, so some microbiome components. So what we did in MyNewGut was to actually try to monitor those diet response, not using the glucose, the glycemia as a marker, but we tried to look at how people respond overall by using their full metabotype. So looking at the metabolic profiles of those people. So can we use actually metabotyping to monitor diet response? Well, it has been shown that in this longitudinal study that was done over 10 years, 22 individuals were followed, and you can see in those plots that every color is a single individual, and every individual is evolves within its own metabolic space. And what they looked at over time is that actually the metabolic space of many individuals was pretty stable. These people, the one that is in red that had a big drop, is because at this point he had an antibiotic treatment. And you can see that it recovers actually quite quickly. And at the following time point, this person was back to its normal metabolic space. So this is very encouraging and shows that using metabotyping, we can actually monitor the individual with a personal metabolism of people over time. Again, that was over 10 years. So how do we do that? Well, in Reading, we have a metabolomic platform that is based on NMR, and this is our main instrument, the 700 megahertz NMR, which is very sensitive, and that we use for the MyNewGut analysis. What we do is basically acquire a metabolic profile into this machine. We use digitalization tools and pattern recognition statistics, and then we identify the metabolites that differentiate between the clusters to understand what's going on into the metabolism. So how did we apply this? Here I'm going to show you a few examples of our diet intervention studies. First of all, I'm going to show you a little bit more details about the high-protein diet studies that François discussed this morning. And so this is a reminder of the design. So if you remember, there was a high-protein diet supplemented in casein or with soy proteins, or a comparator diet that was supplemented in maltodextrin. And what we could see was that actually in this kind of extreme diet situation, we had very clear differences between the groups. So here we showed a very homogeneous response to the diet intervention. For example, in the maltodextrin, a very strong increase in butyric acid. In the maltodextrin group versus soy group. So the soy group was associated with 2-methylbutyrate. That's something that François showed already this morning. And again, we can see that the groups, the clusters, are actually well separated. So the response was very homogeneous. We went a bit further and used those metabotypes to try to understand how the microbiome influenced those. And in feces, we found that the abundance of firmicutes was associated with a very specific metabolic profile. Therefore, we could understand a bit better how the firmicutes overall influence the metabolic ecosystem in feces. Further on, we looked at the correlation between some metabolites and the gut microbiome and identified clusters of bacteria from the Clostridial cluster, actually, that were all nicely correlated with specific metabolites, all associated with protein metabolism. And actually, interestingly, those bacteria have already been associated in former studies. Maybe you're aware of the twins UK cohort where Tim Spector demonstrated that Christensenella, the Christensennaceae family, is a very heritable taxa. And in this study, he showed that it was part of a network with Oscillospira as well. So this is very consistent with what Tim Spector's published already 4 years ago. Moving on to looking at urine metabolites, we also did some correlation analysis with the microbiota, and we found that Butyricimonas abundance, for example, was associated with a very specific profile of metabolites. And I show specifically this one because I found it interesting to illustrate the complexity of understanding those interactions. Because it was associated with phenylacetylglutamine, which is quite a beneficial metabolite, but also very strongly with precursor sulfate, which François explained to this morning that is a quite deleterious compound. So what do you do with that, with this microbe that is actually contributing to both a very beneficial metabolite and a deleterious metabolite? So moving on to other intervention studies, these are the fibers intervention studies we did. And the first one was an Axos intervention study. So in this crossover design, people were either supplemented with fiber, Axos, or with PUFA. And what I wanted to show you is actually the very highly diverse response. In this case, now we're moving to more subtle responses to diet. And when we looked only at this arm, I'm not showing the rest because we identified a little bit of carryover effect and so on. So I'm not going into details, but just showing you that focusing on the first part of the study, the baseline group is here, and you can see that the responders are much more spread in the metabolic space. And those responders were quite peculiar because they had an increase in triglycerides, which is not expected following Axos intake. In the same part of the study, we also looked at the response to PUFA intake, obviously, and here we also demonstrated a very viable response to PUFA intake. You can see that the baseline samples are all nicely clustered together, so they have a very homogeneous metabolic profile. But when we look at the response to PUFA, we can cluster them in 3 different groups. Some people didn't move and remained within the control space, so the PUFA intake had no effect on those people. And then some people moved into this metabolic space, which I call, oops, it doesn't work anymore. I think we're running out of battery. The computer needs to be plugged. Now it works. So these are the one I call the positive responders because they had the expected response to PUFA, which was associated with low triglycerides. But these one moved exactly in the opposite direction. And so I call them the negative responders. They are responders, but they did not do what we expected them to do. So my take-home message here is really that we must be very careful when we do those diet interventions because there is sometimes unexpected responses. And unless we can actually accurately predict who is going to respond properly to those diet interventions, I suggest that we monitor actually quite closely within a few weeks post-diet intervention, whether the people are actually responding in the way we expect them to respond. We did another fiber intervention study, but this one was based on inulin and resistant maltodextrin intake. And again, you can see that there is a huge variability in the response. So this is a baseline metabolic space, and in the dark blue are the post-intervention metabolic space in the fiber group. And as you can see, some of the whole group, the group as a whole, moved towards this, in a quite homogeneous way, towards this metabolic space. And this was associated, as expected, with a reduction. So this was higher in the fiber before the intervention. So these are all the lipids, so the lipoproteins, and some amino acids associated with muscle metabolism. So in this particular study, we actually had a more homogeneous response. Also, it was quite subtle. Just moving on to show you an interesting outcome as well. Although we focused here on the overall response on lipids and a few amino acids, when we looked at the association with the gut microbes, we found that new metabolites were coming out. And for example, for this bacteria, which I cannot pronounce, so don't ask me to do it, the high abundance people were actually also higher quite significantly in glutamic acid. And we're talking about plasma metabolites here. So these people had higher plasma glutamic acid levels. So in conclusion, I think that the level of inter-individual variability of the metabolic response to diet really depends on the magnitude of the diet intervention, because in the high-protein diet, which was quite a drastic diet, we had very homogeneous response. In the second fiber intervention study, I forgot to say that the dose was 20 grams a day, and people had a more homogeneous response. But in the first fiber intervention study, we had more subtle effects, and then some people responded in an unexpected way. So now the next step is really to focus on the integration with microbiota and lipidomic data. This is done in partnership with TNO. And my final word is that unless we have clear metabolic predictors that have been identified before we do the diet intervention, I would strongly recommend that we evaluate the metabolic response of the patients or the volunteers following the diet intervention approximately, I would say, 4 weeks after the initiation of the diet intervention to make sure that they are actually moving in the direction we expect. I would also like you to remember that personalized nutrition needs to be a holistic and a continuous approach. It's not just at one time point. It needs to be done over time. And so if you remember this, I think I've done my job properly today. So final words is about acknowledgements. Obviously, we have a small team in my lab in Reading, and I would like to thank especially Julia Mancano and Joanna Boberska, who have worked a lot on those studies, and obviously the rest of the MyNewGut consortium members, who are too many to be named all here in this slide. So thank you very much for your attention.
Thank you, Sandrine. A very clear talk, clear message. Are there comments? Yes, there are. Please, maybe you can stand up, introduce yourself, and.
Hi, I'm Grace Dugdale from Leeds. I'm really interested in the PUFA sort of work. Just 3 really quick questions. What proportion were negative responders? Did you start by also measuring the omega-3 index? And what exactly did you supplement with?
So I can answer the first question, which is about the proportion of non-responders. That was approximately 1/3, but it's a small study, so I think it has to be validated to have a proper answer. And then you talk about the measurements of the omega-3 index. Yes, I don't think this has been done, or maybe this has been done by the PI somewhere. No?
I'm just wondering if that influences the response.
At least I don't have the information. I can't hear you.
Wait for the mic.
Oh, that was fish oil.
Yes, but do you know which EPA, DHA, do you know specifically?
On top of my head, I would say it was probably a combination of 2.
Yeah.
But I'm not the PI of these studies. So,
Okay,
Joanna knows. Both. I know it was both, but yeah, I don't know in which proportion.